Hello friends, welcome back to my channel Biofric. In today's lecture video, I'll be talking about the basics of DNA and RNA. So all this DNA or RNA are nucleic acids, and the basic unit of the nucleic acid polymer is actually a nucleotide. Now nucleotide has three basic components. Okay, so you have one sugar, second thing here is nitrogenous base, and third thing is phosphate. So all these three components together will make up one nucleotide, and this is the basic unit of the polymer nucleic acid. Nucleoside is sugar plus nitrogenous base. We are not considering phosphate here, so that's nucleoside. Now let's first talk about the sugar. Sugar is ribose sugar. It has 5 carbons here. Now this carbon number 1 that you can see here, it forms a glycosidic linkage, a glycosidic bond with the nitrogenous base. So carbon number 1 is involved with the in the glycosidic bond. Okay, via the glycosidic bond, it will bind to nitrogenous base. And on this carbon number 5, if you look at this particular bond here, it's phosphoester linkage with alpha phosphate. You can have beta later and gamma. But primarily with alpha phosphate, you have a phosphoester linkage. It's not phosphodiester that I'm talking about. I'm talking about the simple phosphoester linkage. Okay, so this is your one nucleotide that we talk about. Now, with regards to this ribose sugar, it can be ribose sugar or deoxyribose sugar. If it is ribose sugar, here you will have OH at carbon number 2. So carbon number 2 is important. Ribose sugar is a part of ribonucleic acid. It will give rise to RNA. If it's DNA, it will be deoxy. There won't be any oxygen. You will just have hydrogen there, and that's deoxyribose sugar. This is the basic thing. Let's talk about this nitrogenous base that's linked to carbon number 1 via glycosidic bond. Nitrogenous bases are of two types, purines and pyrimidines. The ring numbers, okay, here vary. So ring, in the case of pyrimidine, you will have just one ring. Here you have 2 rings here. So under purine, we have adenine and guanine. Under pyrimidines, we have cytosine and uracil. The structure here is very important. Let's get started with uracil first in the case of pyrimidines. So learn this basic structure. If you practice, you will get to know what it means. Like this is NH, so it's a nitrogenous base, right? There'll be nitrogen, and just remember there'll be oxygen at these positions. Okay, that is 2 and 4. So you number it something like this: 1, 2, 3, 4, 5, and 6, like this. Now, if you add one methyl group to uracil, okay, at 5th position, okay, you will get thymine. This is your thymine. That will be red. And in the case of cytosine, everything remains same except for this amino group at 4th position and this extra double bond, which is not here. This is not present. Okay, so just remember, if you practice, you will get to know, and it's important. That's why I'm teaching you this. Now, in the case of adenine, as I said, first understand the purine structure. There are 2 rings. One is basically, it has 6 positions here. It's hexagonal structure. It's pentagonal. The other ring is pentagonal structure. Now, how we will number them? 1, 2, 3, 4, 5, 6, 7, 8, 9. At 6th position, when you have amino group, that's 6-aminopurine, nothing else but adenine. Another way to remember this is if you look at odd positions, 1, 3, except for 5, okay, 5 is exception, 7 and 9. All other odd positions will have nitrogen. It's a nitrogenous base there, right? So that is important. In the case of guanine, here it was 6-aminopurine, right? Here it is. At 6th position, you have oxygen. And at 2nd position, you have amino group. So you can say that it's 2 amino and you can call it 6-oxo, right? And it's guanine. So purine. That's important. Okay. Now, moving further, when we talk about ATP and GTP, which are energy molecule, they store energy in something called as phosphoanhydride. Now, we know the basic structure of nucleotide. So this is the base that is bound to carbon number 1. Here it is OH, that's why it's ribose. If there was no OH and it was just H, it would have been deoxyribose. At 5th carbon, it's linked to alpha phosphate. Okay, and it's specifically linked to alpha phosphate. This is phosphoester linkage. What is phosphoester? This is your phosphoester linkage. Usually ester bond is shown like this, right? In chemistry, we have seen this is phospho. Instead of carbon, you have phosphate. So phosphoester linkage is there. And this particular linkage here is phosphoanhydride. Okay, it's a phosphoanhydride. If there was something like this would have been anhydride bond. This is phosphate here, so phosphoanhydride bond. Now, this phosphoanhydride bond is important for release of energy, and that's where we obtain the energy after converting your ATP to ADP or GTP to GDP. Okay, that's important. Now, the next point is how a phosphodiester bond is formed. Okay, you might get confused. This is phosphoester. Okay, this whole bond here is phosphoester. What is diester then? We have heard of 3-5' phosphodiester linkage. Where does that occur? So we know that there are 2 nucleotides. Okay, now when we need to join them, it's this particular diester linkage. How does that take place? Let's look at this particular image. This is your nucleotide number 1, nucleotide number 2, and now let's consider carbon number 3 and carbon number 5 here. 3' to 5', the link has to be between these two, right? So on this 3' end, you have OH group. Now this O will have a lone pair of electron. This lone pair of electron will make a nucleophilic attack on this particular phosphate, alpha phosphate. Now oxygen will take one bond backwards, okay? It will take, because see, phosphate can form 5 maximum bonds. Now something is attacking this one, and this pyrophosphate, the next step here is pyrophosphate will leave, and there will be new bond that will be formed. Now after that, oxygen can form just 2 bonds, right? Oxygen is already forming 2 bonds, like one is with this carbon number 3 and other one is with hydrogen. Now hydrogen has to leave because oxygen is forming another bond with phosphate, so hydrogen will leave here. So if you look at this image carefully, you will understand. First is nucleophilic attack, second is bonds are taken backwards, pyrophosphate will leave and hydrogen will in the end. So how does your phosphodiester linkage will look like? It looks like something like this, okay? It looks something like this. We have carbon number 3, carbon number 5 here, and there is diester. This is one ester, this is second ester. Diester, can you see phosphodiester, and it's between carbon number 3 and carbon number 5. That's why phosphodiester linkage is formed. Okay, now the next point here is DNA synthesis is always in 5' to 3' direction. Why 5' to 3'? It's clear from here, right? So only 3' OH can attack the 5' phosphate. So let's consider you have 3' OH group. Only this one can attack the next group there, okay, or the next phosphate there. So it has to be from 5' to 3', because 3' will add the next nucleotide. So that's how it cannot go, okay? That's why it cannot go backwards like this. 3' to 5' is not possible. 5' to 3' DNA synthesis is possible, and that's the only truth here. Now, the next thing here is DNA is anti-parallel. Complementarity is important. Why do we get just 2 bonds between A and T, and why do we get 3 hydrogen bonds between G and C? The answer is here. So can you see this? There are 2 places at thymine, and there will be 2 places with adenine that can participate in hydrogen bond formation. In any hydrogen bond, we need to have one H, that is hydrogen, that has to interact, or that has to be in bond with a electronegative atom, and another electronegative atom can be involved, that is Y here, and that's how your hydrogen bond can be formed, right? So here there is N and H. So all these electronegative atoms are fluorine, nitrogen, oxygen, like this we can use here, and hydrogen has to be there. So if you look at this thymine, NH, that can interact with this N, which is Y here, and this is H and X. So that's how you get hydrogen bonds between thymine and adenine, like this. 2 bonds are possible. Between guanine and cytosine, 3 bonds are possible, okay, like that. So now the next point here is that DNA is negatively charged. Why negatively charged? Because phosphate has negative group. In order to avoid repellation, okay, we there is the presence of ions, cations like magnesium or sodium, that will prevent the repellation problem there. Then the next point here is that when we look at any DNA molecule, okay, 1 turn of a bDNA will have approximately 10 nucleotides. 1 turn is also 360 degree. Do you agree with that? So when we divide 360 by 10, okay, we get 36 degree angle. So now which angle am I talking about? Okay, when one pair of nucleotide here, one base pair is present here, the other one will turn like this. So angle between this consequent, okay, one top of the other, we are taking this nucleotide. This angle is 36 degree, like that. You will have one complete turn there. Then we have a diameter that is 20 angstrom, minor groove 12 angstrom, major groove 22 angstrom. Okay, angstrom is minus 10. Sorry, angstrom is minus 10 here, and nanometer, in case of nanometer we have minus 9, right? These terms are important. You can convert angstrom 20, or in the case of nanometer you will write 2 nanometer is the diameter. Next is Chargaff's rule. In the case of Chargaff's rule, Chargaff's rule says that, you know, A will pair with T. So the amount of adenine will be equal to thymine, because they both can form bond with each other and nothing else. And similarly, guanine will form triple bond with C, so the amount will be equal to C, right? Now if I take this particular condition, A is T, so A is 10 and T is 10, so guanine is 15 and cytosine is 15. So if I do the addition of purines and divide it with the addition of pyrimidines, I'll get 1. That's all about the Chargaff's rule, and there'll be many questions that you will see, and you can solve those questions. These are very important questions, okay, based on percentage of purines and pyrimidines. Using this particular rule, you will get to answer those questions. Then the next point here is central dogma. Central dogma is a very basic concept here, and DNA replicates, then there will be transcription, you get RNA, and after translating this RNA you get protein. Now let's talk about another important concept here. Everyone is aware of this, but yet you might get confused, right? So this is the thing that we are doing here. Very basic concept, yet very important. So let's polish them. The mode of replication was a big controversy, I would say. Okay, whether it's conservative as in both the parent strand stay aside as they are naturally, and then newly synthesized strand will be separate. Or semi-conservative said that one template is conserved here, so it's semi-conservative, okay, and newly synthesized strand is, complementary to the template strand. Dispersive was saying something else. It was like one newly synthesized strand will be attached to, it will be complementary to the template and all dispersed. Now, in order to prove that it was semi-conservative, or to find out which model is correct, Messerson and Stark, they came up with a brilliant experiment. Very initially, you should remember that they they cultured this bacteria, okay, in the light isotope N14 isotope, okay, which is a lighter isotope of nitrogen. Now, why do we need nitrogen now? It shouldn't be the question in your head, because we have learned nitrogenous bases are important in the case of DNA or any nucleic acid. So nitrogenous bases, as they replicate here, this nitrogen will get incorporated into their nucleic acid structure, right? So they used this particular thing to design their experiment, and they designed their experiment in such a way that they cultured their bacteria in N14 initially. Now N14 is a lighter isotope of nitrogen, so they got initial result as a light band. What result is this? This is the density centrifugation. Light is less dense, so it will be on top. Moderate is in between, and very heavy isotope, which is N15, which they use later. You will get those bands here. Now, initial result was light band. Then these bacteria, okay, they cultured the bacteria that were grown with light isotope into heavier isotope. Now this is N15. N15, the results that they got were different. They got moderate band, which means that there was one parent strand, the other strand was newly synthesized strand, okay, or it could have been dispersive as well, because it can't be, for sure, it can't be conservative, okay, for sure, because if it was conservative, the parent strand, which is light strand, would have been like this, so light band would have been there, and this newly synthesized strand was heavy strand, right? So heavy strand, so there should have been clear demarcation between that. They didn't get, so they kept open the chances of both semi-conservative and dispersive. Now let's talk about the next step. What did they do? Again, let's go back to the lighter isotope, they said. And now here we know that it was semi-conservative. That time they didn't know. And on growing again in the presence of lighter isotope, this bacteria showed light and moderate density, results. So now, based on this, if it was dispersive, it should have shown moderate again. Why did they get light bands again? How this is possible, getting light bands again? This is because the initial parental strands were still there, okay, and they disrupted, and they formed the complementary newly synthesized strand, again was light. And moderate strands, they were also present here, okay, moderate, because initial isotope that we had used here was N15, so N15 and N14, they became complementary there. That's how you got two light and moderate bands. So they cancelled this possibility. Initially they had kept open both the chances, but now they got clear conclusion that it was a semi-conservative mode of replication that they were looking at. The next point here is DNA replication. Now this is just the beginning, and later in the next lecture video I'll be talking about the detailed topic of this replication. I'll cover that topic. Now here in DNA replication, we need to understand that everyone knows there is DNA polymerase, right? DNA polymerase is there. This is the enzyme that will polymerize, and you can see that there is 5' to 3' direction, 3'OH is here. Now, here we need magnesium for DNA polymerase activity. Why do we need magnesium? It's because in the active site of this DNA polymerase, we have negatively charged amino acids like glutamate or aspartate, or glutamic acid or aspartic acid. It's acid. So there is negative charge on them. Negative aspartate, let's consider aspartate for time being, and negatively charged OH, so OH minus, they both will repel. In order to form a bridge between them, you need this ion. So in any PCR reaction, why magnesium is important is because of this reason. Then there is this thumb, finger, palm analogy that is used. Okay, so thumb here is, this thumb palm analogy is for your polymerase. Okay, so the thumb of this particular polymerase is said to be used for correct positioning. Then fingers will add DNTPs, and your palm here is the beta sheet. So beta sheet of this particular DNA polymerase will hold your DNA strand. So beta sheets of this polymerase, the DNA strand will be held together, okay, on this palm, and then more DNTPs are added like this, and also the correct positioning of this particular strand has to be taken care of. That's done by thumb. So this is a simple analogy that is used. The next question can be asked that DNTPs are specifically added through RNA, although RNA nucleotides are 10 times more. So if you consider overall, the RNA nucleotides are 10 times more than DNA nucleotides, but specifically the polymerase can add DNTPs. Why this is so? Because we know that in the case of your RNA nucleotide, there is OH, and in the case of DNA nucleotide, there is just H at carbon number 2. So this one extra oxygen can cause steric hindrance, and using the same exact difference, there is steric exclusion that is used by DNA polymerase. And DNA polymerase active site has bulky amino acids like glutamate or as I said, aspartate. So it has this ability to discriminate between that OH and H. Now if you mutate this particular active site, and if you change this glutamate to alanine, which is a smaller, not bulky amino acid, then it won't be able to differentiate between DNTP and RNTP. That time it can make this mistake. The next question that can be asked is why only correct nucleotides are added by polymerase. Okay, this is because of optimal alignment, which I already spoke about, how the hydrogen bond is formed, and that's the thing. The next important topic here, which we'll be looking at in the next lecture, but still I have it in my notes, I'll talk about that. So 3' to 5' exonuclease is a proofreading activity. What is a proofreading activity? Okay, it can basically correct the incorrect thing. So whenever you add the nucleotides, A has to be with T. If this is G, for example, it can be corrected. This is the proofreading activity. Now when you have proofreading activity, okay, it will increase the fidelity. It is very accurate, but there'll be the process of polymerization overall will be slow. Okay, and now when I say 5' to 3' exonuclease activity, it is gap filling activity. What do I mean by gap filling? Let's say I have two, this is the gap in between these strands. Now gap filling is the tendency of filling this particular gap at first, and 5' to 3' exonuclease. See here, it will be 5' to 3' if you consider this was also 5' to 3'. When you fill this particular gap, I'll talk about that here now. See, you have 5' to 3' and 5' to 3' like this, right? So you can fill this gap with the new strand. That's fine. And later, this 5' to 3' exonuclease can also remove this particular template, and it will start synthesizing new strand. This is 5' to 3' exonuclease activity. So that is used for Nick translation as well. So gap filling and Nick translation is possible because of this particular activity of DNA polymerase. So I hope this lecture was helpful. It is all basic but yet very important. If you like this video, stay tuned for more such lectures, because I'll be talking about molecular biology and clearing all your doubts, specifically for CSIR NET examination. So bye bye, take care, and see you in the next video.